Okay, let us start the class, guys. I'm audible, right? Hello, students. I'm audible, right? Okay. So let us start the class, guys. Here you can see in the last class, we are continuing the chapter that is sexual reproduction in flowering plant. We are also continuing today also that chapter only. We are completed with agents of pollination. 1 minute. Yeah. We have already completed the agents of pollinations and outbreeding devices, pollen and pistil interaction, right? I think you all are aware of this topic and revised this topic. Then we'll go to the next topic. Okay. Do you have any questions here, guys, in this topic? In this topic, do you have any questions? Okay. So let us study now, guys. Okay. Let us ask some questions. Then we'll move to the today's topic. Okay. The question is, in the last class, we studied the pollen and pistil interaction, right? About that, the question is, the pollen recognition is done by, okay? The pollen recognition is done by, option A, stigma, option B, style, option C, ovary, and option D, pistil. Okay, I think you got the question. The pollen is identified by species specific, right? It is recognized by which one of the following? I'm going to run the poll. Okay, you can answer me in the poll. You answer me which one of the following is the correct. Who is going to recognize the pollen? Okay, this pollen is the specific to this stigma. Who is going to identify? I only told the answer also. Sorry. Okay. Fine. I need answer from others also. Okay. 58% have participated. Remaining, what about the remaining? Okay. I'm going to end the poll, guys. Okay, let us share the result. Here, 75% given the option A, B is 13%, and C is 30%. No one is given the D option. Okay. Yes, guys. The pollen and pistil interaction. When we are talking about the pollen and pistil interaction, this together, all of these, right? Stigma, style, and ovary, we are going to call as pistil. Right. Here, what is happening? The pollen is going to be deposited on the stigma. Stigma, right? Stigma. The stigma is going to be identified or recognized. Okay. So that's why the correct answer is A. Most of them is given the correct answer. Thank you, guys. Okay. Next, another question. A plant produce, this is about the pollination. Okay. A plant produce non-sticky, lightweight pollen grains. Okay. In the large number, and its stigma is feathery. Okay. Which type of pollination it is? In the last class, we studied about the pollination types, right? Wind pollination, water pollination, and insect, right? In this pollination, which is the correct answer? Answer me this very easy question. I want you to answer me in the chat box also, guys. You can see the question. You can answer me in the chat box also, right? What about the lunch, guys? All add lunch. Amiya, HP. HP, you didn't told your name. What is your name? HP and Jermili and Janvi Haveri. Hi, Janvi students. Haveri. Lartu, Lunas, Nandu, Nikita. I want answer from the Janvi Haveri students. Can you tell me which is the correct answer, guys? Very good, Poki. Very good, Terikan. Terikan, can you think once again? Yeah. Terikan, correct? Yes. Yes, Venu. Yes, Sanjushri. What about Janvi Haveri students? Can you tell me your answer? Okay, guys. Correct. The correct answer for this is wind pollination. Yes or no? Very good, Lartu. Yes, Sanju. The correct answer is wind pollination. The wind pollination, the pollen grain should be lightweight and it should not be sticky, right? and the stigma and the stamen should be exposed. We studied that in the last class, right? The correct answer is wind pollination. What about the water pollination? The perianth and other floral parts should be non-wettable here because it is covered by the mucilaginous coating, right? And for pollination by the bee, it should be sticky. We studied, right? Then coming to the pollination by the animals, it will comes under the zoophily. We have not studied that in detail. Okay, then another question. The plant species produces a chasmogamous flower in which androecium and gynoecium mature at the different time. Okay, so if it is maturing at the different time means what is this favoring for? What is this actually favoring for? Autogamy or zerogamy or cross pollination? Harman, Harun, have you answered now? Nanda, have you answered now? This is the correct answer, guys. I'm relaunching the poll. You can answer me in the poll as well as A. Okay, Dharmi is answering. Monica is answering. Terikan given D. Very good. Luna's given A. Imagine, guys, I'm asking the question is don't think in a opposite way. Here, what is happening? No, the plant species that is producing the open flower, chasmogamous flower, open flower. Okay, open flower that is, and androecium and gynoecium is maturing at the different time. They are not maturing at the same time. Remember, if the pollination should be happen or the fertilization should be happen means both should be pollinate at the same time. Then autogamy should be happening. If they are maturing at the different time means what is happening? I'm giving hint also, guys. No, most of them is giving is wrong answer. Okay. So listen here, guys. In the last class we studied, right? Wait, one more given answer. Let us see. Ma'am, it is the dicogamy means it's a phenomenon in the cross pollination. Yeah. So yes, Latuk, that is correct. It is a dicogamy only. But what is happening here? The plant species produces a chasmogamous flower in which androecium and gynoecium mature at the different time. And what is it is favoring for? What is it is favoring for? Is that favoring for self-pollination or for the cross-pollination? In the outbreeding devices, we studied, right, guys? Outbreeding devices. Outbreeding devices, we studied dicogamy, right? In the dicogamy, we studied the chasmogamous flower that is to avoid the self-pollination or to avoid the autogamy. Okay, to avoid the autogamy, the androecium and gynoecium are maturing at different time. Okay, if they are maturing at different time, then only 1 minute. Yeah, they are maturing at the different time here. If they are maturing at the different time means how should the autogamy or self-pollination is going to be happen? Autogamy we studied. Autogamy is nothing but self-pollination. Self-pollination. Yes or no? If the self-pollination has to be happen means both androecium and gynoecium has to mature at the same time. Has to mature at same time. Same time only they have to mature. Then only self-pollination or autogamy is happening. But here what is happening? They are maturing at the different time means they are undergoing the xenogamy. Xenogamy also comes under the cross-pollination. It is called, the correct answer for this is both B and C. Xenogamy means we studied, right? Strange, genetically different, genetically, genetically different, genetically different pollen grain. Pollen grain is going to be deposit on the stigma of the flower. There the cross pollination is going to be happen. Yes or no? That is about the, if it is happening at the different time, the correct answer is xenogamy and also cross pollination. So that's why the correct answer is B and C. Now you understood? While coming to the class, you have to revise, guys. Otherwise, you will not understood anything. Very good, Janvi Haveri. Correct. Lartu, do you understood now? Yes, Lartu, you are wrong. Okay. Just remember the correct answer is the D option. Okay. Fine. This very easy concept, no, guys. Okay. One more chance for you. One more question for you. Okay. Fine. The question is the process where stigma reject the pollen. Remember rejecting. Reject the pollen from the same plant. Remember, rejecting the pollen of the same plant is called as cross incompatibility. Cross, remember the words, okay? Cross incompatibility. Option B, self incompatibility. Option C, self inavailability. Inviability. Option D, self compatibility. Student, if you understood the question properly, without the topic also you can answer to this question. Without the topic also, you can answer to this question without understanding the topic. I launched the poll. You can answer me in the poll. I got so many answers. Very good. Yes, 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 yes. Janvi Haveri, can you think once again? Students, think once again. In the question only they are given. HP, correct. Arun, correct. Yes, Lily. Yes, Vijay. Yes, Jimly. Yes, Nikita. Very good, Nandu. Yes, Venu. Think once again. They are telling the process where stigma rejects. Rejects means we know. Okay. Incompatibility. Right. Incompatibility means what? Rejecting. Pollen from same plant. Same means what? Same means what? Self. Yes or no? Self. Then you got the answer in the question only. Now you tell me which is the answer. Now you tell me, guys, which is the answer. Very good, Janvi Haveri. Yes, Sanju. The correct answer is self incompatibility. Right. In the question only, the answer is there. Okay. Fine. In the today's class, we are going to learn about the artificial hybridization. Okay. What do you mean by artificial hybridization? No, Janvi Haveri. The correct answer for this is self incompatibility. What is happening here is the plant is going to, for example, the flower from the same plant, it is going to reject its own pollen grain because it requires the cross pollination, right? For the cross pollination, this is a condition that is cultivated by the plant. What it will do is it will reject. Okay, if the same pollen from the same flower, if the same flower is there, if the same flower is having the anther and it is having the stigma, this anther is producing the pollen grain, right? This pollen grain will not germinate on the stigma of this flower. Stigma of the same flower. This is same flower. Okay. Why means the condition is called as self, same plant, incompatibility. Incompatibility. 1 minute. I will write it down. That is called as self incompatibility. Okay. Self. Okay. Now you understood, Janvi Haveri. Venu, here self-inviability will not come to this part. Self-inviability means viable means what? It is going to be carry the shelf life, right? The shelf life it is carrying. So here they are not given the a correct statement here. Viability means carrying for the shelf life for some time. Viability. Okay. Inviable means the shelf life will be decreased, or the shelf life it is not having. Okay. Fine. So now let us talk about the artificial hybridization. In the artificial hybridization, first we will talk about, guys. Okay. Don't talk about the fertilization. In the artificial hybridization, fertilization only will come. Later we will talk about that. Okay. Have you seen any different breeds of the dog, guys? Now I need an answer from you. Which are the types of the breeds of the dogs you have seen? Which are the types of the breeds of the dog you have seen? Is anybody is having the dogs in your home, guys? Husky, very good HP. I like Husky so much. Is any other breeds of the dog? Pug. Any other breeds of the dog, guys? German Shepherd, nice. Golden Retriever, very good. Any other thing? Lab, very good. Lab. Yes, Lily, Lab. There are so many, yeah, so many breeds of the dogs are there, right, guys? How these breeds actually come from? Dabur, yes, Dabur, yes, Nandu. So many breeds of the dogs we are seeing, right? How this actually the breeding is coming? Is that happening naturally? No. What we are doing? No. Actually, in the artificial hybridization, we are doing that is called as artificial hybridization. For example, we saw some dog. Okay, we saw some dog that is having a very good hair and it is very cute and that is having a very nice hair. Okay, and good eyes. What we want to do is now we want to cross that dog with the another dog which is having the good characters. Okay, like looking nice and those are having some good character. Now what we will do, now we want the character from dog number one also, character from the dog number two also. Now what we will do, now we will cross this dog, that is female dog from the dog number one and the male dog from the dog number two. Now we will cross both the dogs and the breed is coming, the offspring is coming, no, that is having both the characters, right? That we are doing, that is the artificial hybridization. Okay, so here we are going to call this as the artificial hybridization. Okay, what all our human is making, human are making, this is not natural thing. Whatever the breeds now we are looking, right now we are seeing, those all are not actually naturally occurred. We did actually, we did the hybridization, right? So that's why it is called as artificial hybridization. Okay, fine. Only breeds with the desirable traits are selected as a parent and the mating is going to be performed. Okay, fine. Here you can see, here you can see this dog name is Porgy. Okay, here is this dog name is Porgy. Okay, and here you can see that Welsh Kurgi and this is Pug. Okay, these two is crossed. Okay, for example, the sperm from this dog is going to be put to this female dog. Okay, this pug female dog. Now what will happen? The fertilization happened here in the pug and the dog here the offspring will be, that is called as the porgy. This is as we did, right? The humans did, right? This is called as the artificial hybridization. See, these two dogs are how cute, right? Which dog you like? Anyone, if anyone want to give the answer means you can give me. In these three dogs, which one is cute actually? First one. Lartu liked the first one. What about Jane? We have very students. Pug. Monique also first. Sanjushri also second. Okay, Nandu second. Yeah. Both first and second one is cute, right? Actually, this hybrid wala only not looking good. No one selected this hybrid. Yes, right? Huh. Okay. Fine, guys. This is nothing but the artificial hybridization. Okay. Is anyone having pug in your home? Pug? Is anyone having pug? Third for sake. No. Okay, okay. Ranganayaki Bhargavi. Okay. Fine. No. Okay, guys. Thanks. Now let us now let us come to the topic, guys. Okay. So, and in the plants also we have seen different colors of the vegetables, right? Here you can see are different colors of the cabbage. You can see yellow color, green color, and the violet color. These colors are not naturally occurring in the nature, but these all are actually done, right? These all are actually prepared by the human. It is not colored also, like colored actually. These all are actually hybridized. Okay, and you can see how we are going to select the large size and the colored cauliflower and other vegetables with the varied characters, because you can see now we want the any, for example, any fruit is there means we it is very small in size means, but what we will expect is it should be bigger size, then it will be good. And whenever you see the small grapes, it should be a small grapes, you will see, right, a black grapes will be smaller. Now you can see a different sizes of the grapes are there in the market, right? And the seedless also there, and it is having some different colors also. The grapes have seen that different sizes, right? How this all the things is coming, that is mainly by the hybridization. Okay, and in the rice also you have seen, right, basmati rice, those the rice are actually crossed, right? This is also have done is the artificial hybridizations only. Okay, the rise of the different length you can see, right, that is crossing, the rice also crossing with the a small size rice and the medium size rice, then it is going to be crossed, then we are going to get the a medium sized rice, like kind of thing they are doing. This is nothing but the hybridization. Okay, do you have any other examples for the hybridization, guys? Do you know any other examples for the hybridization as you all seen in the daily life or anywhere? If anyone know means you can tell me in the chat box. Yeah, mule. You have studied in the living. Yeah. In the first chapter, you have studied, right? In the 11th, mule. Mule is the hybridization between. Yeah, rose plants also. Here, yes is given sp3 hybridization. We are not talking about the chemistry hybridization. Yes, Prajwal. Horse and donkey. Yes. Fine, guys. So this is about the hybridization. Okay. Fine. This actually this how this hybridization is going to be done. Artificial hybridization. Let us study, guys, what we are doing, right? First we have to select the parents. Okay, the parents which are having the characters, those are like the dominant characters we can see. Okay, for example, plant one, male parent we are taking. That male parent is having the tall and a rigid stem. Okay, that rigid stem, and yeah, dragon fruit. Okay, okay, Nikita. The plant 12 that we are selecting, that is a male parent, and that is having the trait that is a tall and having the rigid rigid stem. Okay. What we are doing, we are going to collect the pollen grain from that. Okay. Then we are going to select the female. Okay. The female also should have some characters, right? That also should be dominant in some character. What we are doing, that female should produce a large fruit. Okay. It will produce a large fruit we are selecting. Okay. Now what we are doing is, here two characters are there. One is tall and rigid stem. The male is having, and the female is having the large fruit size. Now we are expecting the progeny. That progeny is having both the tall and rigid stem, and that is producing the large fruit. Okay, because parents are having that characters, right? That's why the offerings produced from these parents are having the tall and rigid stem and with the large fruit size. Okay, so this is how we are going to do the fertilization. That is the artificial hybridization is going to be do. Okay, fine. Here you can see, guys. In the plants we are doing means we have studied about the yeah, yes, Ranganayaki. Ranganayaki asked a very good question. What if the negative characters also will come means, right? That hybridization is going to be fail. Yes or no? Yeah, sometimes it will happen, Ranganayaki. We can't assure that it will get the positive characters only. If it is in the animal, we can think, we can do the some examination, then we can analyze the characters, and then we can do the hybridization. But what happen in the plants? No, in the plants it will be in the multiple numbers. We will do the hybridization. No, we can't guarantee that actually in the plants. Okay. After some time only, we'll come to know that all the characters are correct, all the positive characters are there or not. But in the plant, the negative characters will be like in the limited. Okay. In the plant, it is difficult actually. Good question, Rangnaiki. Okay. In the plants, we know that unisexual flowers are there and the bisexual flowers are there. Right. You know that unisexual flowers are mean a single flower. Okay. Single flower that is having either ovary, either gynoecium or the uh, gynoecium or the androecium, right? Ovary or stamen. It is having, right? Either one of them it is having unisexual flower. In that actually cross pollination is easy or difficult? I want a question from you guys. Yes, thing unisexual flowers having the single reproductive organ, either the male organ or the female. In them, the cross pollination is easy or difficult? Here, just imagine we are doing the hybridization means it is cross pollination only, right? In the unisexual flower, it is easy, or in the bisexual flower, it is easy? Easy. Very good, Nikita. In the unisexual flower, the hybridization is very easy. That is, that too, artificial hybridization is very easy. We will think about that, how it is going to happen. Okay. Yeah. And bisexual flower, you know that single flower has both the male and female part, and complete flower it is called as. Okay. I will tell you guys here. Okay. See here, in the unisexual flower, it is having either male or female. In that cross pollination will be very easy. What we will do is, see here, this, for example, this flower is there. Okay. 1 minute. Okay, I will tell you this. First, you make a note of this. In the next diagram, I am going to tell you. Okay. First, we will do the emasculation. What do you mean by emasculation? Means removal of anther, removal of stamen. Remember, removal of stamen. We are going to cut the stamen. We will cut the stamen. Okay. And what do you mean by bagging? Bagging means we are going to cover the stigma. We are going to cover the stigma. Okay. Why we are going to cover the stigma means no any other pollen grain should will come and bind to the stigma, right? So that's why we should bag that. Okay. First understand the definition, then I will tell you. Very good, Ranganaiki. Very good, Sanjur. Correct. With the polythene bag. No. Actually, some filter papers will come. Through that, we are going to cover pa. Nikita. For that only, some filter papers will come. Through that, we are going to be covered. Okay, not polythene. Some paper kind of cover will come. Through that, we are going to cover. Okay. Here you can see, guys, what is happening. First understood what is emasculation. Emasculation means a removal. We are cutting out the stamen. Stamen consisting of the anther that is going to produce the male gamete. Remember. Okay. Then what do you mean by bagging? In the bagging, we are going to cover the stigma. Okay. I think you understood the emasculation and bagging. Can you give me thumbs up if you understood the word emasculation and bagging means? Can you give me thumbs up, guys, if you understood emasculation and bagging means? Or give me your emojis, guys. Yes, your emojis. Fast. What about Janvi Haveri students? Yes. Yes, Carlson, with the butter paper. Yes, Pa. For the bagging, we are going to use the butter paper. Thank you, Clarkson. Yes. Ranganai, okay. This is your emoji, right? I thought you were raising the hand. Okay, fine, guys. So now let us come to the bisexual flower. Okay, just remember in the unisexual flower, we are not going to do the emasculation, just we are going to do the bagging. Okay, then we'll come to the bisexual flower. Bisexual flower, where it is producing both the male and female part is there, where the androecium and gynecium both are there. We are going to remove the anther from the female parent. What we are doing? We are going to remove the, in the bisexual flower, we are going to remove the, yes, we are going to remove the anther from the female parent. Okay. Female parent, anther we are going to remove. That we are going to call as an emasculation. Okay. And we are going to cover the stigma of that female parent with the butter paper, that is called as the bagging. I will make you to understand clearly. Just you understood this concept now. Very good, Ranganayaki. Why we are going to do the bagging? To prevent the self-pollination or prevent the contamination. Why we have to do the bagging, guys? For example, if you in the unisexual flower, you see in the unisexual flower, if we are not the emasculation, we are not doing. Okay, we are doing the bagging. Why we are doing the bagging means till we are depositing the pollen grain of our interest, till it should not get pollinated from the some other pollen grain, right? It should be not get pollinated from the some other pollen grain, right? The other pollen grain should not be deposited on the stigma. For that reason, we are going to do the bagging. We are going to cover. Okay. Otherwise, the others will be get uh, deposited on the stigma and it gets polluted, right? So that's why we are doing the bagging. Contaminated. It is going to be get contaminated. Okay. The bagging will be done for that. The emasculation we are doing because the self pollination, to avoid the self pollination. Okay, fine. Let us understood by the diagram. Okay, see here. Okay, so here you can see in the bisexual flower we will see first. Okay, bisexual flower. The flower is having both male and female part. Here you can see the flower is having the female part and this flower is having the male part, right? What we are doing is the pistil part and also stamen it is having, right? The pistil from the flower of the desired female parent. We are taking the, see here, we are taking the pistil of the female flower, female parent. Okay. Now what we are doing is, now that female parent is having the male stamen also, right? We are going to cut out that. We are going to remove that. Okay, remove that by the scissors. By using the knife, by using the scissors, we are going to remove the stamen. Okay. Now we remove the stamen, right? The emasculation of the stamen. We removed, right? Now what will happen? This flower is having this. We have did the bagging, right? We have did the bagging, right? We did the bagging. Now what we will do is, now we are going to deposit the pollen of our interest. We have chosen some male parent, right? We have chosen the some male plant which is having the dominant character, that is having the good stem and that is having the tall stem, right? Now we are going to put that, we are going to collect the pollen grain from that male and we are going to deposit it on the female parent, where that female parent is used to produce a large fruit. Large fruit. Okay. On the stigma, we deposited. After deposition of the pollen grain, we are going to do the bagging again. We are going to do the bagging again. Okay. Because the some other pollen grain should not deposit and it should not be going to grow right inside the it should not going to be interact, right? So that's why we are going to cover the stigma to avoid the contamination of that stigma. Okay, here you can see this is how they will do the covering. Okay, this is a mustard plant. Okay, 1 minute. Do you understood this, guys? After dusting the pollen grain from the desired male parent, the flower is bagged. The flower will be bagged to avoid the cross pollination from the unwanted pollen. The unwanted pollen should not come and bind on the stigma, right? So that's why we are going to do the bagging. Do you understood this now? In the bisexual flower, how the artificial hybridization will be done? Did you understood, guys? Give me thumbs up or any emojis if you understood means. What is this, guys? You all are in home. Now only you had your lunch and now the students are in JNV, had lunch. Are you getting sleep, guys? No one is answering in the chat box. Very good, Lartu. What about others? Aram se sleeping up. I know you all are studying and you all are watching, right? Okay, guys, fine. Just I'm joking, okay? Fine. Okay, this is in the case of bisexual flower, guys. Okay. So now let us talk about the unisexual flowers. The flower is either having the male or female parent. Okay. In that, what is happening? Here, you can see we are not going to do the emasculation because the anther is having in the different parent and the female is having only the female part, right? Not the male part. So that's why there is no case of self-pollination, right? Yes or no? There is no chance of self-pollination. Why we have to remove the emasculation? Why we have to do the emasculation and why we have to remove the anther? Yes or no? So that's why in the unisexual flower, okay, in the unisexual flower, we don't require emasculation. Emasculation is not required. Okay. So what we are going to do in the unisexual flower is we are going to select a male parent. We will collect the pollen grain from the male parent and we will dust that pollen grain on the stigma of the female flower which we selected and we are going to do the bagging. That's it. Now, after the bagging, you know that after the fertilization, what are the things is going to be happen, right? It is happening inside the bag. Do you understood this? Here you can see pollen grain from the flower of the desired male parent and pollen from the desired male parent dusted on the stigma of the desired female parent. Okay, we are taking here from the male parent, we are taken the pollen grain and we have dusted here on the stigma of the female flower. Now we are going to do the bagging. Okay, after dusting the pollen grain, we will do the rebagging or the bagging to avoid the contamination. This is about the artificial hybridization, guys. This is completed. We explained both unisexual flower and also the bisexual flower. And here is a question for you. Okay. In the unisexual flower, which of the following correctly describe the hybridization? Okay. You can go through the question. 1 minute. Cross-pollination is easy and only emasculation is performed. Self-pollination is easy and only emasculation is performed. Cross-pollination is difficult and only bagging is performed. Option D, cross-pollination is easy and only bagging is performed. Read the options correctly, then you go for the answer. Read the questions correctly. Very good, Tanjivur. Very good, Rangnaiki. Very good, Venu. What about others? I'm launching the poll. You can answer me. 1 minute. Yeah. I have relaunched the poll. You can go through that poll. Think once again. Just remember, guys, in the hybridization, artificial hybridization, we are doing only cross-pollination. There will be a no self-pollination. The cross-pollination is easy and is only emasculation is going to be doing the unisexual flower. What I told, emasculation is not at all required, right? Yes or no? I told emasculation is not required. See here, 40% given A, B is 13%, C is 13%, and 33% D. Go through the last slide, what I told you here. Guys, in the unisexual flower, the emasculation is not performed, right? Only bagging will be done. Why no emasculation? Because both anther and stigma or both androecium and gynoecium are in the different plant. There is no self-pollination is happening. So that's why we are removing. That's why there is no emasculation is required. Okay. Okay, the correct answer is option D, guys. Cross-pollination is easy because only bagging is performed in the unisexual flowers. Okay, fine. So let us talk about the double fertilization, guys. Okay. First, let us talk about the fertilization. Then we will understand the what is double fertilization. Okay. Double fertilization is a characteristic feature of angiosperm. Oh, sorry. Is it Nikita? Okay, you can answer me in the chat box also, pa. Okay, but I'm launching the poll. Okay, Nikita, fine. Okay, now let us talk about the fertilization, guys. Okay, in the plant. Now you see, what do you mean by fertilization, guys? Can anyone tell me what do you mean by fertilization? Fertilization. May I get answer from any student? What about HP, Lartu, Monica, Nandu, Jainvi Haveri, Nikita? What do you mean by fertilization? The fusion of male and female gamete. Simple. Right. The fusion of male and female gamete we are going to call as fertilization. Right. And syngamy also. Syngamy. We will call fertilization as syngamy also, guys. Syngamy. Syngamy. Okay, fine. Let us study. Now, in the post-fertilization event, in the microspore mother cell, the microsporogenesis has happened. By the microsporogenesis, we got the male gamete. Yes or no? We got the male gamete. Pollen grain we got. Pollen grain we got. Yes or no? This is a male gametophyte. In the pollen grain, we got the gametes. Gametes we got. That gametes is nothing but the generative cell. Generative cell is acting as the two male gamete. Two male gamete. Generative cell is acting as two male gamete. Now let us go to the female. Megaspore mother cell. Megaspore mother cell undergoes the megasporogenesis. Then it gives the embryo sac, female gamete, right? It given the female gametophyte. The female gametophyte, we call that as the, 1 minute. Yeah. The female gametophyte that we call as embryo sac. Embryo sac. This embryo sac have the egg, right? This embryo sac is having the egg, and this one of the generative cell is going to be fused with egg. One of the generative cell is going to be fused with this egg, right? Can you tell me whether the gametes are haploid or diploid? Gametes are haploid or diploid, guys. This generative cell is also haploid and egg cell is also haploid. In the embryo sac, we have seen antipodal cell, synergid cell, egg cell, central cell, right? All are haploid only. Remember, all are haploid only. Okay? Fine. Now you can see these haploid cells will fuse to form the zygote. This is nothing but the fertilization. Right? This is what we studied in the fertilization. Very good, Rakshita. Very good, Lily. Very good, Sanjur. Okay? Now, the male gamete and the female gamete is ready. Now let us see how the fertilization is going to be happen. Okay. Sorry, I thought to remove this. Fine. Okay. 1 minute, guys. Okay, let us understand this. See here. What do you mean by double fertilization? Double means two times. Two times, right, guys? Double means two times. Here, the fusion is happening two times. Okay, two times. So that's why it is called as double fertilization. Okay. So here, what will happen? The pollen grain is having the two gametes, right? That is the two male gametes. That is nothing but two generative cells are there, right? These two generative cells, it is going to be brought by the pollen tube to the different cells of the same gametophyte. Okay, 1 minute. Okay, here I will tell you guys. 1 minute. So here in the double fertilization, what will happen means the pollen grain is having pollen grain is there, no? Pollen grain. In the pollen grain, there will be a three cells are there, guys. Three cells, right? In the three cells, there will be a one vegetative cell, one vegetative cell, and the two generative cell is there. Two generative cell, right? This two generative cell is acting as a gamete, okay? And here two gametes are there, right? Two gametes. And one vegetative cell is helping in the formation of the tube cell. Tube, pollen tube is going to be formed, right? There, it is going to be act as a tube cell. Okay, it is not going to be fused. Okay, the two gametes, one gamete is going to be fused with egg. Okay, one gamete is going to be fused with egg. And another gamete, there is a central cell is there, right? Central cell that is having the two nuclei, right? The one of the gamete is going to be fused with this central cell. Okay. Now you can see there will be a three nucleus will be there, right? So that's why this is called as endosperm, and this is called as 3N, 3N. Okay. Primary endosperm nucleus we will also call as pen. Pen means primary endosperm nucleus. Okay. Do you understood this? Do you understood this? Now let us go to the part. Okay. 1 minute. Okay. So here you can see, guys. Okay, I will explain first this thing and then we'll go to the next. Okay. In the fertilization, guys, based on the pollen tube entry, based on the pollen tube entry, there will be a three types. If the pollen tube is entering through the micropylar end, see here, if the pollen tube is entering through the micropylar end, it is called as the porogamy. It is called as porogamy. Kindly make a note of this. I will erase this later. Kindly make a note of this. If the pollen tube is entering to the micropylar end, it is called as porogamy. Pollen tube. Okay. If the pollen tube is entering the egg cell through the chalazal end. Okay. The pollen tube entering the egg cell through the chalazal end, it is called as chalazogamy. Chalazogamy. Okay. If the pollen tube that is entering through the center, you can see this is a center, right? Integument. Through the integument, that is called as mesogamy. Okay. Kindly make a note of this. Micropylar end means porogamy, chalazal end means chalazogamy, and through the integument means it is called as mesogamy. It is based on the entry of the pollen tube. It is based on the entry of the pollen tube. Okay. Kindly make a note of this. I will remove this. Okay. Let us understand here, guys. Okay, what is happening here? The fusion of the two male gametes brought by the pollen tube to the different cells of the same gametophyte. Okay. Ranganayaki, actually, I don't know. I will check and I will tell you, Pa. Okay? Is that okay, Ranganayaki? In the tomorrow's class, I will tell you. Okay? Fine. So here you can see, guys, what will happen. This is the pistil, right? This is a female part. Here you can see this is consisting of the stigma, style, and the ovary, right? This part is ovary, right? In this ovary, there are ovules are there. How many ovules are there? One, two, three, four, five, six. Six ovules are there here, right? You can see this is the first ovule. You can see this is the one ovule. Okay, we are going to check the fertilization here in this ovule. Okay. Now the pollen grain is deposited on the stigma. Okay. Now this pollen is specific. It is compatible. Pollen grain is compatible. Now the pollen tube has been germinated. The pollen tube has been germinated and it is entering. It is entering the ovule. It is entering the ovule through the micropylar end. Micropylar end. Through the micropylar end, the pollen tube, it is entering to the ovule. Can you tell me if it is entering to the micropylar end means what is that called as? What is that called as? It is entering through the micropylar end means. Now only I told. Porogamy, chalassogamy, mesogamy. Very good, Rakshita. Very good, Rakshita Talwar. Correct. Porogamy. Very good. Yes. This is called as a porogamy. Now, the pollen tube is formed from vegetative cell, right? Now, this pollen tube is carrying the two gamete. Which are those two gamete? The two gamete is nothing but generative cell, right? Two gamete. The nuclei from the generative cell is going to be carried by this pollen tube. You can see here. Two tubes are there. Two, right? Sometime what will happen, oh, guys? If the pollen tube is carrying only one generative cell means, during the pollen tube formation, the another generative cell is going to be formed by the mitosis of the generative cell. I told in the last class, right? Gymnosperm will be, most of the gymnosperm, 60% is going to be shed at the two cell stage. Two cell stage means one is vegetative cell and one is the generative cell, right? For the double fertilization, we require, in most of the angiosperm, double fertilization require the three cell stage, right? That is nothing but one vegetative cell, two generative cell, right? Totally three cell. Sometime if it is shed at the two cell stage means here the generative cell will again it will undergo the mitosis during the pollen tube formation. During the pollen tube formation, it will undergo the mitosis. Okay, remember that. Now we have the two male gamete here. Now it is entering to the micropylar end, and it is here you can see it is entered to the ovule. It is entered to the ovule now. What will happen now? We will see. In the last class, we discussed it, guys. Who is going to guide the pollen tube entry into the egg? Now what will happen? No, this pollen tube is going to be entered through the ovule and the nuclei will be there, right? Nuclei, two gametes, male gamete. Now one of the male gamete is going to fuse with the synergid. It is going to fuse with the synergid first and that is going to be burst out. Okay, remember the one male gamete is going to be fused with the synergid first, then this male gamete is going to be, this synergid is going to be burst out. Now, this male gamete, the male gamete released from the synergid, right? That is going to be fused with the central cell. Okay, male gamete is not fused there. Okay, that is not fused with the synergid cell. That male gamete just enter the synergid because the synergid and the filiform apparatus is guiding the pollen tube entry, right? The filiform apparatus and the synergids that guide the pollen tube entry, the gamete, that male gamete enter to this, that will enter to the synergid and synergid will burst out. That male gamete is going to be combined with this central cell. Now there are two cells are there and again male gamete is also fused. Now this is called as 3N. 3N, that is triploid, and it is also called as pen. Pen means primary endosperm nucleus. Primary endosperm sperm nucleus. Nucleus. Now this 3N, it is undergoing to form the endosperm. It is going to be developed into the endosperm now. Okay. Now the another male gamete is there, right? Now another male gamete, it is going to be fused with the egg. It is going to be fused with the egg, and this is going to be form the zygote. Zygote. Okay. This zygote is going to be form embryo. Embryo. Okay. You have to remember which is going to be first development. The endosperm is going to be first developed, then embryo is going to be developed. Do you understood this, guys? The endosperm going to be first developed, then embryo. The embryo is going to be developed from the zygote. The zygote is 2n, right? 2n, right? It is going to be developed into the embryo. Do you understood these things? Do you understood this thing? This thing we will revise tomorrow. Okay. In the tomorrow's class, we will revise once again the double fertilization. Okay. Fine. Yeah, you can give me a class feedback. I'm launching the feedback, guys. You can give me your class feedback. We'll complete this chapter in the tomorrow's class. Okay. This is the first chapter, right? Some important points and some important concepts we have to understand slowly. So that's why I did slowly. Now the coming chapters and all the things will go fast. Okay. Or this much slow is okay for you all. Yeah, Nandu, bye. Yeah, thank you so much, guys. We'll meet in the next class. Lartu, I didn't saw you in the class only. You didn't answered anywhere. You didn't talk also in the class. I remembered you, Lartu. Be there in the class. I will explain you. Okay, 1 minute.